Hi, everyone. Thank you so much for joining our webinar today on culturing kidney organoids. So our speaker today is Dr. Ben Juliar, who is an assistant research professor at University of British Columbia. Ben comes with a really impressive background in organoid generation, and he's going to be sharing some of his insights and tips for differentiation and maintenance of kidney organoids. But before I go ahead and pass it off to Ben, as always, I just want to share a few slides introducing protein tech. So, Protein Tech is a leading provider of life science reagents. The company was founded in 2001, and at that time, it was a primary supplier of antibodies and immunoassays. And because we have been in the game for over 2 decades, we have a really large portfolio of antibodies that cover about two-thirds of the human proteome. We also offer ELISA kits and IHC kits, as well as cell separation kits and antibody labeling kits. Our company then later expanded to include other product lines. So we also have now our humankind branded cytokines and growth factors. And all our proteins are going to be expressed in human HEC293 cells so that you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins, which ultimately increases their bioactivity. We also have our Chromatec branded nanobody based reagents. So, nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, making them ideal for several applications such as immunoprecipitation, immunofluorescence, especially a super resolution microscopy, as well as protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics, and they manufacture antibodies conjugated to oligonucleotides for single cell RNA sequencing. So, as a company that is driven by scientists, we are here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. For convenience and testing, most of our antibodies are also offered in 20 microliter trial sizes. We also know the importance of product validation, and we offer open access validation data for our customers. Whenever we are testing new products, we validate with relevant sample types, appropriate experimental conditions, and using knockout, knockdown models whenever possible. Also, as antibody experts, we have in depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform, which we use to manufacture our recombinant antibodies. With 7 inventory sites and manufacturing and research on 3 different continents, we are able to offer next day delivery to many areas in the world. So, as a company, we do measure our success by our customer scientific success, specifically by helping them publish impactful research faster. So, this graph here shows that over our company history, our products are being featured in more and more publications. And as of now, we have over 350,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also just really quickly want to highlight that as a company, we do strive to make the searchability of our products on our website as user-friendly and informative as possible. So we've recently launched some new updates on our website to help our customers select the right antibody. So this includes our unique 3D epitope viewer, which allows you to interact with epitope structures and explore antibody binding sites, so you know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need, such as our immunogen region selector toggle that allows you to filter for antibodies generated from your region of interest. Additionally, you no longer have to filter through all the variations of an antibody from the search results. You can now view all the formats of an antibody, including conjugations and buffer formulations, on just one page, which ultimately streamlines your process for antibody selection. And then lastly, I want to highlight that we have recently launched an AI-powered search tool on our website called Able. So Able allows you to directly chat and ask for product recommendations, help with experimental design, and access relevant validation data and resources. So for example, you can ask Able, what antibodies can I use to characterize my kidney organoids? And Able will tailor a reagents list for you so that you don't have to spend countless hours searching for the right antibodies. So, this AI powered tool is the first of its kind and was designed with our customers in mind so that you can all focus on your discoveries and not reagent selection. So, we are really proud to be the first in the industry to launch this. So, if you do have some time, please do check it out. You can access the bot by scanning this QR code right here, or you can simply just go onto our website and you'll find Able right next to the search bar. So, with that, I would like to go ahead and introduce our speaker for today, Dr. Benjamin Juliar. So, Ben is a new assistant research professor in the Division of Nephrology at the University of British Columbia. His work focuses on using stem cell derived kidney organoids to model disease, study inflammation driven injury, and explore regenerative therapies. With a background in tissue engineering, biophysics, and translational kidney research, Ben is working to establish a research program integrating vascularized organoids for applications in drug discovery and bioartificial kidney development. So, with that, I will go ahead and pass it off to Ben. Thank you so much for being here today and for offering your insights to us.
Sure thing. Yeah, one sec while I share. All right, is that full screen?
Not yet. Yes, now it is.
Okay, great. Yeah, thanks, Frida. So as she mentioned, I'm a new assistant professor at UBC. I just finished my training, my postdoctoral training at the University of Washington in Seattle under Benjamin Friedman, who is a pioneer in kidney organoids. He helped establish one of the first protocols to differentiate them. And I haven't even updated my LinkedIn yet. It still says postdoc. I need to get around to that. But like Frida mentioned, I've been using kidney organoids for disease modeling. And I'm going to give you some tips on how to differentiate them. Honestly, kidney organoids can be very finicky. And a lot of times, if your differentiation doesn't work, then you just really can't run your experiment. So it's really important to do the optimization for the organoids and really make sure that you are starting from the best material possible beforehand. As a general overview for this talk, I'll be talking about, well, one, just like kidney structure, if anyone is unfamiliar with kidneys, how to differentiate stem cells into kidney organoids, like the general protocol, tips for optimizing robust organoid formation, including considerations for the media and how to treat your stem cells, troubleshooting tips for variability in your organoid culture or low viability of your organoids, characterization of organoid morphology and the typical markers we use to demonstrate that the organoids are mature, as well as some recent research to improve the maturity and make the phenotype more physiologically relevant. And then best practices for maintaining organoids over extended culture periods, which we're starting to work on and is a relative or was starting to work on, and is a relatively newer consideration for organoids. And then I'll take a question and answer. So for those who don't know the general structure of the kidney is broadly. Let me get my. I'm not sure. Frida, does a laser pointer show up? Or I'm trying to find the laser pointer.
I can
Well,
see your mouse, but the laser pointer, you should have an
oh, okay, yeah,
option.
mouse is fine.
Okay.
And so the kidney is generally is broadly split into the cortical region and the medulla. And the cortical region contains the glomerulus and proximal and distal tubule systems. The glomerulus has a really important function in the primary filtration of blood. So it lets small molecules pass through, like roughly 60 kilodaltons or smaller. So that retains most proteins, but nutrients and salts pass through. And then the proximal convoluted tubule system is really the workhorse of reabsorption. So it reabsorbs nutrients, electrolytes, most water. And then the tubules pass down into the medulla and the loop of Henle. And this portion is really important for salt and water balance, as well as concentrating urea. And then pass back into the cortical region for the distal tubules, which has additional functions in water and salt balance, and then goes to the collecting duct, into the hilum, and that passes ultimately to the bladder. So for kidney organoids, the organoids are essentially generated by mimicking differentiation within the body. And so we mimic mesoderm induction, and then within the mesoderm, there's the anterior intermediate mesoderm as well as the posterior intermediate mesoderm. And so for kidney organoids, at least the protocol that I've been working with, we only get cells of the posterior intermediate mesoderm. And that's important because we're only getting cells that form the upper portion of the nephron. We're not actually getting any collecting duct cells. So the anterior intermediate mesoderm forms into the ureteric bud, which then forms into the collecting duct. Whereas the posterior intermediate mesoderm turns into nephron progenitor cells, stromal cells, and vascular cells. So these are the cells that we ultimately get from our differentiation protocol. And so that ultimately becomes the glomerular cells. So that's podocyte-like cells, endothelial cells. We think we get the parietal epithelial cells. So those are the cells that form the Bowman's capsule on the outside of the glomerulus. We get proximal tubule cells and distal tubule cells. It's kind of hard to see whether or not there's any loop of Henle cells. Not sure about that. And then we don't get collecting duct cells in my differentiation that I'm familiar with. There's a lot of work ongoing to try to get ureteric bud cells, ureteric bud organoids, and then combine those with nephron progenitor cell organoids and get a more mature structure, but that's still very much ongoing. And so our kidney organoids, we get cells from the metanephric mesenchyme, which then differentiate into the nephron progenitor cells. And we get podocytes, tubules, and generally endothelial cells. But we don't really get like a defined Bowman's capsule structure with parietal epithelial cells surrounding the glomerulus generally. We've had like some suggestive results, but it's not like a consistent thing. And then we definitely don't get any of the ureteric bud, so we don't get any of the collecting duct epithelia cells. So getting good organoids really depends on starting with healthy stem cells beforehand. I can't emphasize that enough. Like how you treat your stem cells and how you maintain them is essential for getting good organoids. And the actual the cell line that you're working with, like for every cell line, like you need to make like small tweaks to the protocol. So there's a few stem cell lines that I've worked with, and I'm sure you can make organoids from more stem cell lines. Friedman Lab is banking a lot of patient-derived stem cells to try to get organoids growing from patients into personalized medicine. But the most common cell line is the WTC11. So that's like our most consistent cell line. And we use that a lot for gene engineering and for if you're trying to knock in proteins or knock out proteins or do GFP tagging. We pretty much always use a WTC11 background because they're one of the most consistent cell lines that we work with. and that's an induced pluripotent stem cell that's commercially available. Sorry. We also use H9 embryonic stem cells. So that's another commercially available cell line. Generally, the H9 cell lines, they don't grow as long or as fast. Like you can't passage them as much as the WTC11s. I'm not sure exactly like the physiological reason for that. But the H9s, they work well, and it's nice to have to test your protocol in both like iPS cell lines as well as embryonic stem cell lines. So they're also like a good standard to work with. A lot of researchers use the H9s. I've found them to be a little bit less consistent than the WTC11s. And then another commercially available cell line that I've worked with is this 1016 SEV-A line, also an induced pluripotent stem cell line. I've also found this one to be less consistent than the WTC11 line, but it's good in organoid experiments in general to do your experiments with several cell lines, like if you have the capacity for it, because sometimes you'll have a differentiation that works for whatever reason in one cell line and not in another. So like just kind of trying in several cell lines is generally good practice. It's also like reviewers love to see that you've done your experiments in several cell lines just to add some robustness to your experiment. So I definitely recommend having several stem cell lines to do experiments with. For whatever reason, if just in your hands one cell line might not work, but another will. And I mean, I don't think there's any good known scientific reason, but if you're a stem cell researcher, you'll definitely be aware that there is just variability like that, and it's hard to get around except for trying a bunch of different things. And so generally speaking, these stem cells, when they're healthy, you see like really tightly packed cells in these colonies with nice, well-defined edges, like you can see here. In practice, you'll generally see some surrounding cells that have differentiated into some unknown phenotype. They kind of look fibroblastic. In my opinion, we haven't really characterized exactly what they differentiate into. But generally, if you're doing like releaser passaging, so releaser passaging helps lift off the colonies all together. That usually leaves behind a lot of the single cells, and you can help clean up the single cells if you're doing releaser passaging. As opposed to the other type of passaging we usually do is accutase, which then suspends the cells as a single cell suspension, and you get some of these stromal cells. And so we usually do releaser passaging just for routine maintenance of our stem cell lines, and then accutase passaging if we want to do seeding with an accurate cell number. So single cell suspension, you can quantify your cell yield and do and plate cells at specific densities for your organoid experiments. When we're first thawing our stem cells, we generally keep our stem cells frozen in freezer S freezing medium. I'm sure you could probably use DMSO and FBS as well, but we've found freezer S to be very good at helping maintain the stem cell characteristics. So we like to stick with it. Again, like the more variables you can remove. So like FBS, there's just batch to batch variability. So like the more defined the media, generally speaking, the better. Although freezer S is kind of expensive. So I guess that kind of depends on your lab and just how much money you have available. For our culture, we always coat our plates with Geltrex. And so this is a reduced growth factor basement membrane, and it's qualified for human embryonic stem cells. I would definitely recommend the growth factor reduced. It's more expensive, but if you start adding growth factors, then that can induce stem cell differentiation. And then you always want to make sure to passage below 80% confluence. Like once the stem cells get too confluent, then that also induces it induces a combination of quiescence where they stop growing as fast, as well as differentiation. So it's important to make sure that they have enough space to keep growing and passage them pretty often. So they'll generally double every day. And depending on what media you're using, so MTs are one, you'll need to feed them every day, which is kind of a hassle. An M-teaser plus, you can feed them every other day. It's the growth factors. I think it's the FGF9, I want to say, in the M-teaser plus. That's been engineered so that it's more stable. You can feed every other day or 3 times a week if you double up on the media over the weekend. And I definitely, from a workflow standpoint, prefer the M-teaser plus. But with the caveat that you need to be really careful, like before going into the weekend, to make sure that your cells are at low enough confluence that they're going to make it through the weekend. And because the M-teaser plus is the growth factors are engineered to be more stable, I find that the stem cells tend to grow faster. At least I think that's why they grow faster, is because the growth factors are generally at a higher concentration and more active. So you do need to be more careful about really checking that the confluence is staying low enough, using the M-teaser plus. So that's very important for the stem cell maintenance. If you let your cells overgrow, I'd probably recommend just tossing them, to be honest. If they hit 100% confluence, just thaw a new aliquot. Before you start doing a lot of experiments, I'd also recommend having like a large bank of stem cells that are all identical, like 20 some aliquots that are all just like identical and lower passage, so that you know consistently, like, okay, when I freeze this when I thaw this alicot, it's going to take however many days to get to confluence. And then when you thaw a new aliquot, I'd also recommend passaging it at least twice before you do an organoid experiment, so that you can get out like dead cells, or make sure that the cells that are healthy end up dominating the culture. And then, yeah, if it gets confluent, I toss it out. Or if the cells lose their tight colonies, so they look like more dispersed like this, more like cobblestone morphology instead of like tight small bodies, or more fibroblastic around the edges, I would toss those as well. Yeah, like organoids, they take forever. So starting with good stem cells, I can't emphasize it enough, is really important. There's a few organoid differentiation protocols that all were established around the same time, like I think 2015. I can't remember exactly, but that's when a lot of the seminal work happened. Like I mentioned, I'm coming out of Bino Friedman's lab. So our protocol starts with the pluripotent stem cells, so either embryonic or induced. You plate the stem cells onto Matrigel, and then after a day, sandwich them so that you've got basement membrane on both sides of them. And then that induces them to form spheroids, so that essentially polarizes the stem cells. And then we add Kyer, which is a GSK3 inhibitor, which activates Wnt signaling and induces differentiation towards mesenchyme. And then we use B27, which is a supplement actually originally used just for maintaining neuronal cell culture. But also helps induce kidney organoid differentiation. Honestly, that's a bit of a black box. I think it was more of a serendipitous finding than anything. But it makes for a relatively simple protocol. I think the Friedman protocol that I'm familiar with is more simple than most other protocols, and that's something that we like. And so then we get some neuronal cells, we get endothelial cells surrounding, we get these glomerular tufts, and then distal tubules, or I mean, and proximal tubules. I just mean general tubules. Sorry. So tubular structures kind of associated with the glomerular tufts and endothelial cells surrounding. Other protocols are a little bit more complicated. They often differentiate to nephron progenitor cells and then dissociate them. Which adds an extra step, but it also allows you to freeze down nephron progenitor cells and bank them for later. And then you can either plate those into U-bottom plates where they form spheroids and then organoids, or people have also worked to put them on a transwell plate, which has like a different, where you can basically have an interface between the top reservoir and bottom reservoir. And these studies have been trying to see if you can get like filtration. So a little bit more detail on our kidney organoid differentiation, as well as representative images. So when you plate your cells, you definitely want to make sure that you have single cells. You don't want clusters. The spheroids should arise from, I mean, these little cavitated spheroids, or we call them donuts, after the matrigel sandwiching, generally arise from just a single or maybe two stem cells. So having your like an actual like single cell suspension is very important when you're doing your accutase treatment and plating. We include a ROCK inhibitor during plating, which increases the viability of the cells. Like I mentioned, the basement membrane extract, so matrigel, induces polarization of your stem cells after they've grown. And then you get these little, these donuts, we call them. And then we let the donuts grow for a day, and then we do a 36-hour Kyer treatment. In practice, it's generally okay if this goes for more like 40 or maybe 42 hours. But it depends on the cell line you're working with. And we do generally 12 micromolar Kyer. I guess I forgot to write that down. But we also titrate the concentration of Kyer when we're developing a new protocol for a given cell line. So like that's one of like the key considerations is whether we include Kyer. We also include noggin, depending on the cell line, which is a bone morphogenetic protein inhibitor. So that helps force the cells towards the mesenchyme lineage. And then I've also occasionally used a TGF-beta-1 inhibitor to help with differentiation. And so then after the Kyer treatment, we just keep the organoids in the RPMI or advanced RPMI plus B27, and then they develop into kidney organoids. So at day 10, you start to see these, it's kind of like it almost looks like broken mud, but there's these islands of renal vesicles. All the scale bars are 100 microns, so you can appreciate it's maybe 200 or 300 microns across. And then they eventually develop into the tubular organoids, where you can really see these nice tubular structures. And then these are sometimes maybe 400 or 500 microns across. And so the most standard staining that we do is podocalyxin for the podocytes, and it's very typical for the podocytes to have these kind of like a tuft-like appearance, maybe several tufts around the outside. And then the body of the organoid has the proximal and distal tubules. But generally speaking, we just stain for the proximal tubules with LTL, lotus tetraglobulin lectin. And then the endothelium grows in a network around the outside. And then there's also usually a thin layer of stromal cells. I guess not very, kind of not showing because it's only like a single layer, and just the way I thresholded it, the DAPI is a little bit light, but surrounding the organoids, there's generally like a sparse monolayer of what we assume is generally like a stromal like lineage. We don't generally characterize the cells surrounding the organoids very well, but when we do single cell RNA-seq, there's always a large portion. I mean, the mesenchymal like cells like dominate the signal. Honestly, most of the cells in the culture are a stromal cell of some sort, or so we kind of assume that the cells surrounding it are stromal cells. So some of like the culture conditions, or some of like the tips that I would recommend for your differentiations, are definitely make sure that your media is good. Yeah, so you obviously need stem cells that are healthy coming into the differentiation, but because the differentiation is so dependent on the actual environmental factors, it's really important that you follow like the supplier's recommendation for your media storage. So like, when you're warming, and then M-teaser is expensive. It's like $200 a bottle. It's ridiculous. And so, but I would also recommend, like, if it's old, like more than 4 weeks old, I mean, your time is more expensive than a bottle of M-teaser. So like, or yeah, so like M-teaser for culturing the stem cells or like the organoids at the beginning, and then your advanced RPMI with the B27. Also, B27 is pretty expensive. That's usually something like $80, 70 or $80 a vial. So your culture media is also expensive. But for either one of those, I would recommend following the supplier's recommendation of like only 3 to 4 weeks at 4 Celsius, and then only heat up like what you need to actually feed your organoids at the time, or your stem cells at the time. Like, don't heat up the whole bottle. The more you like heat and cool it, the faster the growth factors are going to degrade. I already mentioned the considerations for the M-teaser 1 versus M-teaser plus. M-teaser 1 is kind of inconvenient, but it does force you to pay attention to your cells every day. M-teaser plus is more convenient, but it's kind of easier to forget about your stem cells and let them get overconfluent. Your differentiation also highly depends on the seeding density. So like I mentioned, like making sure that you have actual like single cells when you're plating your organoids is really important. And then depending on the cell line you're working with and like how fast they grow, that also has a factor on how many cells to plate. The WTC11, they grow faster. I also think that they're more robust than the H9 cells. I think part of the reason why we need a higher density plating density for the H9s is just because a lot of the cells don't adhere. And then I'll usually do a range of seeding densities for every single differentiation. So like if I want to have a full plate of organoids, I'll actually plate 3 plates that have a different seeding density for each one, centered around a seeding density that I know works well. So like if you're just starting to do your organoid differentiations, you might want to do a wider range of seeding densities. Here I've listed kind of my recommendations. If you're doing a 96 well plate, it's more like 300 to 500 cells per well. But yeah, making sure that you do a range of seeding densities is really important, especially when you're optimizing your protocol. And then the Kyer dose also is really important. I mean, that's what initiates the differentiation. So getting that just right is really important. If you do too high of a concentration, then it's toxic for the cells. If it's too low, then you don't get efficient differentiation. So again, that's an important consideration when you're working with a new cell line or when you're just trying to optimize a protocol, is to do a range. We usually do a range from, like if I'm doing testing a new protocol, I usually do like 8, 10, and 12 micromolar for a variety of cell seeding densities. So like all of the permutations of that. And then I'll also include like plus minus noggin. And so then with that, you end up with, I don't know, what is that, like 18 different conditions, even if you have a 3x3x2. And so it can be a lot of things to test at first, just to get your protocol optimized before you are even really doing experiments. And then I just wanted to mention that, yeah, B27 is a proprietary blend, but B18 is published, and it's what B27 was formulated off of. So there's a lot of components to it, and there's definitely a general interest in moving away from like proprietary blends or kind of these more complicated blends and moving more towards like a well-defined media. And so there are protocols that are out there that are fully defined. And if you're just getting into organoid differentiation, then it might be worthwhile like trying our protocol versus other protocols. But because there's so many components in B27, there's a lot of lot-to-lot variability. So in practice, what our lab will do is get like 6 different lots, and test them to see which one is best. So we'll do like 6 different lots with often like a variety of cell seeding densities, and try to figure out which one is best for given cell lines. And then stock up like a 2-year supply of B27. And so that's also a bit of a headache, is the lot testing. But definitely worthwhile, because sometimes you just have a lot that doesn't work very well. So some troubleshooting tips are to make sure your stem cells are healthy. I think this is more for iteration. Make sure you've got your fresh media and growth factors, that everything is stored properly. Actually, how you're physically pipetting makes a big difference. So organoids are very delicate, especially after the Kyer treatment. So like gently pipette, do your mycoplasma tests. They're hard to detect, but they will affect your differentiation. Do your Kyer and cell seeding titration. Take live microscope images, really helps if you're getting poor differentiations. Like take an image every day, even just like a representative image, so that you can go back and like pay attention that you're getting all the structures, and that can help figure out like, okay, at what step are things going wrong, at least like qualitatively. And then keep a log for every differentiation, like break down what are your seeding densities, what's the passage number of the stem cells going into it, any details that you notice along the way, just to like help make sure, because sometimes it's really frustrating if you don't have a log and you're trying to figure out like, okay, why hasn't my differentiation worked like 3 times in a row or whatever. You need to have a reference to go back to like try to troubleshoot. So the general markers we use to demonstrate that the organoids are mature. So for podocytes, you can use synaptopodin, nephrin. So synaptopodin and nephrin are often pretty lowly expressed in the organoids, to be honest, but they're there. They're not like super strong stains, so you might need to use like a relatively high concentration. And then ZO1 is generally pretty highly expressed. The PEC, so the parietal epithelial cells, they form the Bowman's capsule generally. We've had some suggestive results that they're there. You can use claudin 1, which is a tight junction protein. They're nephrin negative, and then you get nuclear Pax8. And then for proximal tubules, they are LTL positive, epithelial cadherin negative, and cubolin positive. And then the distal tubules are generally LTL negative, epithelial cadherin positive, and cubolin negative. And then the tubules, they have this very distinct tubular structure as well. So you can often tell even just from DAPI staining where the tubules are. I wanted to mention that some of the future work that's happening, so this is out of the Lewis lab, trying to get the vasculature perfused. People are putting organoids into hydrogels, and then the vasculature can grow out of the organoids into the hydrogel, and that allows for the potential for perfusion. But again, like, nobody has yet gotten actual like filtration of the organoids. The organoids are still more like a structure. Like you can only do like structural assays with them, I should say, rather than actual like functional assays so far. So that's still like a big research push in the field in general. And then also, yeah, getting a collecting duct system integrated with the perfused vasculature and the tubules, like that's all still, I think people are working on it, but you know, that would be a huge breakthrough if someone makes that to work. So I also want to note that as far as longevity goes, that it's typical for the organoids to start to degrade over time. So they're generally mature after about 23 total days in culture, maybe like 21 to 23 days. So like the whole differentiation protocol takes roughly 3 weeks, and then you can start to do experiments with the organoids. And it's typical. So these are GFP podocalyxin organoids. So they're the podocyte tufts. You can see these are live images. And this is the same organoid at maturity, and then a week later. And I think you can probably appreciate that the podocyte tufts have gotten a bit smaller. And that's actually just kind of typical for the organoids. At least in our protocol, pretty much all protocols have like a defined endpoint that's around the 3 to 4 week mark, and I don't think anyone's published actually like a long-term culturing of kidney organoids for like longer, like several months at a time. But quantitatively, we also see like this decrease in podocalyxin, and part of it, I think, is because the stromal cells, they tend to grow faster than the epithelial cells, so they kind of like overgrow. They just kind of consistently overgrow and start to dominate the culture, and so then the epithelia starts to wane away. But the mature organoids, you can see here that it's lasted about a week. It would probably last another couple weeks before it just like completely degraded. The endothelial network that we form though is generally like pretty mature at the same time as the organoid. So like you're a mature organoid, and a week later you should have endothelial cells to start, and then they should like persist for at least a week. Afterwards, they may actually continue to grow because they're kind of part of the stromal fraction. And then I want to mention this work out of the Cleaver's lab, where they've actually gotten indefinite growth of just the tubular structures by supplementing media with tubular enhancing growth factors shown here. And they can basically take their organoids, dissociate them into tubules, and then consistently passage them. I can't remember for how many passages. I think it's something like they did like 10 or 20 or more. I just showed 3 passages here, but they can just consistently passage their tubules. So I think conceivably you could probably do a similar thing with the organoids with the right media that enhanced the growth of the epithelium, but not the stromal fraction. But again, that's something that is like ongoing work, something that I've been interested in and done some preliminary work, but nothing published yet. And I guess with that, that's all of the points I wanted to make. So kind of in summary, the organoids, they only, at least the organoid model that I'm working with, just only models the nephron progenitor cell lineage. But there's emerging research on ureteric bud and combining ureteric bud organoids with nephron progenitor cell organoids to get more mature systems. There's a variety of differentiation protocols depending on your needs. And yeah, I'd recommend maybe trying a couple. The stem cell sourcing and quality is definitely a very important concern, as well as your media and how you're actually like physically pipetting. And then the markers of maturity are mostly like structural and like what protein expression. There's not really like functional markers of maturity like perfusion and filtering yet. And then you can usually do your organoid tests for like maybe 2 to 4 weeks after maturity, but be careful that the organoids start to degrade. And then there's ongoing research to improve the longevity of your organoids. So with that, I can open up to any questions. I think we got about 10 minutes.
Great. Awesome. Thank you so much, Ben. That was a really wonderful talk, and I definitely learned a lot, especially since I've never worked with organoids before. So it was some really great information for me. Before we begin the Q&A session, I do just want to share a couple more slides. Let me go ahead and switch this up. All right. So I just want to take a few minutes to highlight some of our products. So specifically our humankind growth factors and the advantages that they offer that make them ideal for the differentiation and the maintenance of your kidney organoids. So in Ben's talk, he emphasized the importance of making sure you optimize your media and you properly feed your cells. So at Protein Tech, we currently do offer 80 RUO and 58 GMP grade cytokines and growth factors, including the essential proteins that are needed for culturing kidney organoids. So one of the key advantages of our humankind proteins is that they are authentic human proteins. So this is because they are derived from human HEC293 cells. So they have native post-translational modifications and protein folding in order to optimize the biological activity. So this is often lost when proteins are expressed in other systems like E. coli. Because of their native nature, we have observed that humankind proteins have superior bioactivity and better stability, which means you'll get enhanced results and healthier cultures. And specifically, we have also seen advantages in cell expansion and differentiation protocols. Because of our quality manufacturing process, you can also be confident that our proteins will be animal component free, xeno free, endotoxin free, and tag free. I also just want to touch a little bit more on why the expression system matters and why we put so much emphasis on the fact that our proteins are derived from human cells. So here on the right, we are looking at a comparison between our humankind TNF alpha compared to an E. coli derived TNF alpha. So for proteins expressed in non-human expression systems, some of the pain points are things like lack of authentic post-translational modifications, high endotoxin levels, improper protein folding, and lower bioactivity. Additionally, with E. coli-derived TNF-alpha, we see that they tend to be expressed in monomers with no post-translational modifications. Whereas in our humankind system, you can see that our TNF-alpha is actually expressed in trimers, which is going to be the most active form of TNF-alpha. And we also have the proper post-translational modifications and proper protein folding that would essentially mimic what you would observe in human TNF-alpha. So basically, you can be confident that the protein that you are exposing your cells to is equivalent to what you would observe in a human system. So here again, we're showing the benefits of our expression system in regards to protein assembly and post-translational modifications. So on the left, we're comparing CHO expressed activin A versus our humankind activin A, and you can see that humankind activin A has more active dimers compared to CHO derived activin A, which has more of the inactive precursor. And then on the right-hand side, we are comparing our humankind beta cellulin versus E. coli-derived beta cellulin. And here in this plot, you can see that the humankind beta cellulin is highly glycosylated, whereas the E. coli-derived beta cellulin is not. And again, emphasizing the native post-translational modifications and the biological activity that you can expect with a human expression system. So on the previous slide, I showed you some data for activin A, which of course is going to be an essential growth factor for kidney organoids. Another key growth factor is, of course, FGF2. So here I want to highlight specifically the superiority of our thermostable FGF2. So we understand, the headache of having to continuously replenish FGF2 in your cultures. This can not only be time consuming, but can end up costing quite a bit of money. And that's why our team engineered this thermostable FGF2 for enhanced stability in culture media without having any sort of alterations to its biological function. So the comparison of our thermostable FGF2 to the NIBSC standard shows that our FGF2 is 12 times more active, degrades 3 times slower, and can maintain pluripotency even with a 2-day feeding cycle. So overall, with this thermostable FGF2, you are not only getting a product that performs better, but also lasts longer, which ultimately saves you time, money, and the headaches that come with culturing organoids. And then lastly, we understand, the importance of lot-to-lot consistency for the reproducibility and reliability of your organoids. So here is an example of our PDGF beta beta and activin A. And we observed that between 3 independent lots, the activity and the purity was nearly identical. So that kind of ensures, the data reproducibility and quality that you get with these products. And so then just to summarize our offering, so as I mentioned earlier, we do offer over 80 RUO and 58 GMP grade cytokines and growth factors, including the essential proteins for culturing kidney organoids, such as activin A, BMP4, BMP7, FGF9, FGF2, and more. And then additionally, in addition to our growth factors, we also offer an expansive portfolio of unconjugated and conjugated antibodies that can help you characterize your organoids. So here we're showing an example of our LRP2 polyclonal antibody that was used for IF in fixed human kidney tissue. And then as an attendee of today's webinar, you will receive 2 follow-up emails. One will have the recording of this webinar as well as a link for you to request a free sample of one of our humankind recombinant proteins. So you can select from this list of these 4 right here. You will also receive a separate email, which will contain a certificate of attendance. And we do ask to please allow 1 week for this follow-up email to be sent. If you are eager to request your free sample, you can also go ahead and do that now by scanning this QR code. So with that, I would like to go ahead and start the Q&A session. If you do have any questions for us after the Q&A session is over, you can always feel free to email us at events@ptglab.com, or you can reach out to Ben directly at bjuliar@prostatecenter.com. And if you liked today's webinar and you want to learn about our upcoming events, please do follow us on our social media. And if you want to look up, any past webinars or any resources that we have available, you can always ask our AI chatbot, Abel, so you can scan the QR code there and begin chatting with him. So with that, I can go ahead and start the Q&A. There are a lot of questions I saw that were coming in. So
Yeah, I've started typing some responses.
okay, great. Awesome.
Um,
So if you've covered some of these, I've had some written down, but if you've covered these, we can move on to a different question. So one of the questions was, can you use uh, thiazine instead of Y27632 as the ROCK inhibitor, and what is the concentration that is recommended to be used?
yeah, so for the rock inhibitor, that's 10 micromolar. I suppose you could probably use other rock inhibitors, yeah, if it has the same function, but I actually replied to that one and I wasn't sure uh that specific rock inhibitor. There's several available. So again, I would recommend just testing it in your own hands. If it works, then it works. Is kind of how it goes. All
Okay.
right, yeah, next question.
Great. So you mentioned that at day 3, you add or don't add noggin and SB. What is the difference with or without the addition of these two growth factors? If I am not wrong, the original paper did not use them.
Yeah, so those are both to help like force the cells into the mesenchyme lineage. And so some cell lines they need like an extra push and some don't. So these are just like additional growth factors that are well, one noggin's a growth factor, the SB compound is an inhibitor that can help get stem cells down the right lineage. So, but again, that kind of like just depends on the line that you're working with, and there's just additional considerations for protocol optimization.
Great. Another question about that protocol. So do you change the media between day 3 and 10 and between day 10 and day 21? If yes, how many? And if no, do you add growth factors?
Yeah. So before the Kyer treatment, the media is changed every day, and up until that 36-hour time point. And then we switch to the advanced RPMI. It's every other day until day 10, and then every 3 days or 3 times a week until maturity. So generally speaking, you want to change your media every other day would be a kind of like best practice. But if you skip a weekend after the differentiation's over already happening, that's generally fine. And that's like from a practical standpoint, what I would usually do.
Perfect. Thank you. Do you have any recommendations on how you can optimize media composition? So, for example, if you want to do multi-organ modeling or similar, and you need to change the original media you use to accommodate the second organoid, how would you go about changing and optimizing that media composition?
Yeah, so the most straightforward thing is just to look at what growth factors. I mean, so generally speaking, the basal media for most cell culture is very similar. You'll have some trace elements that are in some media but not in others. So I would just generally just kind of add everything that you might need if you want to do culture 2 different stem cells. So this is something that we come into for especially when we're trying to enhance our vascular growth. So it's like, you've got your vascular endothelial growth factor media and your organoid media. And it's like, okay, what is the minimal? The organoids are more finicky. Well, you want to figure out like which organ is more finicky. Um, and then just supplement the minimal amount of growth factors from like the less finicky system, let's say. So endothelial cells are generally more consistently formed. And so then if you just add like VEGF and ascorbic acid to uh, your differentiation media, then we get like really nice vasculature to form. Um, if you're doing it for another organ system, I would probably have a similar approach where it's like, okay, which one is the more robust differentiation? Probably initiate the differentiation separate, and then afterwards co-culture in a media that has the essential growth factors from both systems. Um,
Great.
yeah, or even just like titrate the media 50-50. I mean, that's like a simple way to like, I don't know, just get a rough idea to start.
Awesome. Thank you. I think we have time for maybe a couple more questions. I know we're at the top of the hour, but didn't have a whole lot of time left over, so I just want to make sure that we answer some more. Um, so one of the questions that just came in is, does a shaker help for organoid formation, or do you think that could disturb the cells and the structure formation?
Mm. Yeah, I'd probably recommend against a shaker. You definitely want to be delicate. And most of these systems, at least the way we're doing it, like the wells are small enough that like things are diffusing quickly. Um, I guess maybe a shaker could be useful if you have some sort of setup where you need to increase diffusion of molecules or oxygen, but um, I would say generally probably advise against it.
Okay, great. And then I think this is a really good one to end on. So this person is asking, at present, do you think we can use kidney organoids to investigate kidney disease, for example, fibrosis and other injury, or is the application still limited to kidney development? What about the use of kidney organoid conditioned media to investigate inter-organ communication?
Yeah, so disease modeling is the whole point. So yeah, that's spot on. Fibrosis in particular is of interest. In practice, it's hard to get the cells to actually like generate a lot of ECM that like actually mimics fibrosis. I mean, you can definitely get the cells to upregulate genes associated with ECM and maybe get a little bit more deposition, but getting like a genuine fibrotic phenotype, I haven't seen. But in theory, I think a fibrosis phenotype is doable. And yeah, actually something that I'm interested in. So maybe, yeah, I'd say that and I'm sure probably some people are working on it. That's a hot topic. In practice, yeah, you can also model genetic diseases. So the Friedman lab is like their bread and butter is this cystic model where if you engineer a PKD, polycystic kidney disease gene, then the organoids they form these large cysts. The model that I was using it for is studying cytokine-induced cell death. So then you can look at cell-dependent differences in cytokine treatment. And I saw that endothelial cells preferentially died with interferon due to pure apoptosis. So I was using that to study its contribution to kidney disease. Um, and then like cystinosis, where you delete the cysteine gene, then you get cysteine accumulation in the organoids, and we're working to try to show that that has a phenotype. For disease modeling, always getting a robust phenotype is the hard part, though. You can often see like cellular differences, and a lot of times people present the phenotype as okay, you get like you engineer whatever gene, and you're getting differences in gene expression. But it is often hard to see microscopically like a distinct phenotype. But yeah, that's the whole point of the kidney organoid model is for disease modeling.
So is it safe to say that you foresee this being kind of the future of disease modeling?
I'd say it definitely complements mouse models. So it's a nice like intermediate scale. Um, I think from a practical standpoint, it's important to have like every level of disease modeling. So like monoculture experiments are much faster and you get like cleaner results, like less variability in the actual metrics. Organoids are like medium scale as far as time goes, and effort. And you can screen a lot of conditions, but then you get noisier results. So if you don't have like a really clean phenotype, then it's hard. And then mouse models, you have the whole system, or other animal models, you have like the whole system, but it's more of like a black box, and it takes a lot of effort, and it's really expensive to screen many conditions. So every level is important for disease modeling, and it really just depends on your application. And in practice, we try to bring them all together and use them appropriately.
Great. Awesome. Thank you so much, Ben. As I mentioned, that was a really wonderful talk. I know the comments that are coming in also mention the wonderful lecture. I do just want to, again, put up this last slide with the contact information. I know we did not get to everyone's questions, so I apologize. But if you want to directly reach out to either us or Ben, you can feel free to do so. You can always contact us at events@ptglab.com or contact Ben at bjuliar@prostatecenter.com. So with that, I think I can go ahead and end the webinar. Thank you everyone for attending, and I hope you have a great rest of your morning, afternoon, or evening, depending on where you're tuning in from the world.